My name is Lindsay Morton. I'm a scientist at the National Cancer Institute in the Division of Cancer Epidemiology and Genetics. And I've been working on lymphoma and studying the causes of lymphoma for over a decade. I'm an epidemiologist, which means my job is to study what causes diseases and look at the patterns of diseases that occur in different people and in different places and over different time periods. For the last 4 years, I've been leading the InterLymph NHL Subtypes project, which is a collaboration among investigators in North America, Europe, and Australia, where we combine data from 20 studies in order to look at the risk factors for all NHL subtypes. So there are 20 studies that are participating in the InterLymph NHL Subtypes project. And each of these studies was run out of different academic institutions or the National Cancer Institute in the United States. And within each study, patients were recruited who had lymphoma, and all of these patients were asked to fill out a questionnaire. And the questionnaire asked them about their history of different exposures or different patterns of behavior. And then individuals who are similar ages and a distribution of men and women were also asked to fill out the questionnaire. And so we have compared the rates of different behaviors or exposures in the patients with NHL to patients who do not have NHL. And that's how we understand whether or not something is a risk factor. So we have two goals in the InterLymph NHL Subtypes project. The first goal is to comprehensively look at the risk factors by NHL subtype. And we're including all non-genetic risk factors that we have data on. So we have information on the medical history factors, such as autoimmune diseases, hepatitis C viral infection, allergy, and other atopic conditions. We have data on the family history of cancer, so in first-degree relatives, parents or brothers or sisters of the patients themselves, and other exposures, such as lifestyle exposures, history of cigarette smoking, alcohol consumption, obesity, occupation, and history of living or working on a farm. So a second goal of the InterLymph NHL Subtypes project is to see if we can understand more about whether the risk factors are the same or different for different NHL subtypes. In comparing the risks among the NHL subtypes, we've been really struck by some similarities and some differences in the risk factors across subtypes. So one example is autoimmune diseases. Autoimmune diseases have long been recognized as increasing the risk for non-Hodgkin lymphoma, but it really appears to affect different specific autoimmune diseases really appear to affect specific lymphoma subtypes differently. So there are a couple of subtypes that are particularly associated with autoimmune diseases, such as marginal zone lymphoma and diffuse large B-cell lymphoma. And you see many fewer associations between autoimmune diseases and other lymphoma subtypes, such as follicular lymphoma or chronic lymphocytic leukemia. There are some other risk factors that appear to affect very few subtypes, like cigarette smoking, where we see risks for follicular lymphoma and peripheral T-cell lymphoma, but not for most of the other lymphoma subtypes. So although cigarette smoking doesn't appear to cause many types of lymphoma, it certainly is still a very important risk factor for cancer, particularly lung cancer and pancreatic cancer. So for the InterLymph NHL Subtypes project, we've worked really hard for about 4 years to combine the data from these 20 studies, and then to conduct the centralized analyses and prepare the results for publication so that we can share them with scientists and with patients. And the hope now is that we'll be able to combine this effort with the ongoing genetics studies that InterLymph is doing. And ideally, what we would be able to find would be risk factors that perhaps are only in certain individuals who have a genetic susceptibility. And so although we can look at our risk factors in the full patient population, which is what we're doing right now, we do hope that ultimately we would be able to better understand those risk factors and exactly how they cause lymphoma by combining all of the data on medical history and family history and lifestyle with all of the genetics data.